Okay, thank you for the very kind introduction. My name is Ramona Woitek, and I'm an academic radiologist at the University of Cambridge. And a big part of my work is on ovarian cancer imaging. And those projects are what I'm going to talk about now. I hope I can move on my slides. Yes. So in the beginning, I would like to outline a little bit what the challenges are in ovarian cancer and high-grade serous ovarian cancer. It's the particular type of cancer we work on, and this type of cancer has the highest mortality of all gynecological malignancies, of all malignancies in women, and a big part of those women with this cancer are diagnosed when the cancer has already spread throughout the abdomen, and when we find cancer deposits in multiple sites in the abdomen. And that is also reflected in a low 5-year overall survival rate, ranging between 10 and 40%, depending on the stage at which the disease is diagnosed. What is extremely important for these patients is to completely remove the cancer during surgery. And on this plot, you can see that the survival curves for these patients are actually different depending on how much disease is left after they have undergone surgery. And when in cases where the disease is very extensive or where there is a lot of bulky disease in anatomic locations, where it is difficult to resect the disease. These are the cases when neoadjuvant chemotherapy is often recommended. That means that patients undergo several cycles of chemotherapy before they undergo delayed surgery. This is a very successful concept because it allows to shrink down the disease and to facilitate the complete resection of the disease. But in those patients who do not respond very well to the treatment and where the tumor doesn't shrink, we cause a delay in surgery, for the surgery of several months. And that is, of course, a problem. And the question we wanted to address with one of our projects was whether we can predict a patient's response to neoadjuvant chemotherapy at baseline. And we created a multi-omics data set to work on. Our data set was comprised of an internal data set that we used for training, and we had a holdout validation data set, and then we had a completely independent external validation data set for all of these patients, just under 140. We had CT scans at baseline, and then also after they had finished their neoadjuvant chemotherapy. And we did manual segmentations of the whole disease burden throughout the whole abdomen and also in other sites. And we combined, so for the images, we computed radiomics, and we combined the radiomics with the clinical data that were available for these patients with CA125, which is a blood-based biomarker that is very often assessed in patients with suspected or diagnosed ovarian cancer. And then we also included circulating tumor DNA assessments that were done for these patients. And with this multi-omics data set, or we fed this data set into an ensemble machine learning framework, and we basically wanted to predict, as I said, a patient's volumetric response to neoadjuvant chemotherapy. And what we found was that when we used clinical data and CA125, the blood-based biomarker, the addition of CA125 did not really improve the predictions a lot. The correlation between the predicted response and the measured response on the images didn't improve a lot when we added CA125, and the mean squared error didn't decrease much. So the addition of CA125 really didn't help a lot. But once we added radiomics into the features parameters for this predictor, we can see that the error of the prediction decreased quite significantly, both in the holdout validation set and also in the completely independent external validation set, while the correlation between the predicted volumetric response and the measured volumetric response increased. So this is what we wanted to see. For the internal data set, we also had circulating tumor DNA, and that didn't add much on top of the radiomics. So we can see radiomics are quite important for these predictions. We can only obtain those radiomics if we have segmentations of the whole of the entire disease burden in those patients. As I said, we developed this on a cohort of around 140 patients, but of course we would like to validate this in a larger data set, ideally a multi-center data set, so that we can show that these predictions are robust, and we can apply them to also a very heterogeneous data set coming from multiple institutions. To be able to do that, we need the infrastructure, and here the XNAT repository plays an important role, and we also need segmentations, and the segmentations are often the bottleneck for these projects. So we are now collecting imaging data from the ICON-8 trial. The ICON-8 trial was a trial that looked at different dosages and different timings of a certain type of neoadjuvant chemotherapy that was given to patients with ovarian cancer. And a large number of those centers that took part in that trial are now contributing to building this multi-center data set. And they are basically helping us by uploading their anonymized CT scans to the XNAT repository in Cambridge. And of course, they are not allowed to send us any CDs. We only accept uploads of fully anonymized data sets. We help them with the anonymization, and we think this is a very good way of building such a data set for the validation of this predictor, and also for developing new tools. The segmentation, as I said, is often the bottleneck. We spent many hours doing these segmentations manually. A certain level of training is also required, and that's why we are also working on developing an automated segmentation tool for particularly for multi-site ovarian cancer. And Thomas Budenkotter, who is a PhD student, is working on adapting the NNUNet to our needs, basically, and he has a data set of, again, CT scans prior to neoadjuvant chemotherapy and after neoadjuvant chemotherapy. He uses our manual segmentations as the ground truth, and this is a nice comparison of a case with the manual segmentations on top and the automated segmentations shown just below. And for the disease that is located in the pelvis and also in the omentum, these are the two most common manifestations of the disease. For these locations, the automated segmentation works quite well, and it has the potential to save us a lot of time. And of course, only with these extensive segmentations are we able to compute radiomics. And this is an overview of how we implemented the automated segmentation in the XNAT repository and the OHIV viewer. And this is all only possible thanks to Lorena Escudero, who is leading all efforts related to the XNAT repository in Cambridge. So what we do is basically we and our participating centers, they upload their anonymized CT scans to the XNAT repository. Lorena embedded the automated segmentation tool within the NVIDIA Clara framework so that we can access it via the OHIF viewer and we can let it run on our uploaded images. We get those segmentations. We can review them using the OHIF viewer, and we can also adapt or change them if we feel they are not correct. And I'm going to give you an example of this. This is a short video. I hope I can start it. So this is how we access our images on the repository. I think you're familiar with that. And under the mask button, we can access the NVIDIA AI-assisted annotations. There we select segmentations, and we start our segmentation tool. And it takes a few minutes before we can see the result, and this will be a result for the pelvic disease that is fully automatically segmented. We can go through the stack of images. We get we can display the segmentations, and if we are unhappy with some parts of the segmentations, we can modify them, and this is a huge improvement. Even if there are tiny parts that we are not 100% happy with, of course, this speeds up segmentations a lot. And what we are planning now is we would like to see whether radiologists with different levels of training are still able to distinguish between automated segmentations and manual segmentations, and we're going to do this via a Turing test. This test will be implemented in the OHIF viewer. This is still work in progress, but we are going to present again the 3D stack of images to the readers, and we're going to ask them whether they think they are looking at an automated segmentation or a manual segmentation. We would also like to know what they think the clues or hints are, and that's how we would like to learn more about those automated segmentations. And of course, we hope they won't be able to distinguish between the two. So I showed you that radiomics are very important in making those predictions of response to chemotherapy, and I would like to speak a little bit more about why they are important in other ways as well. I said earlier on that ovarian cancer is often diagnosed when it has already spread to different locations in the abdomen. And that is because it arises in an area that is not very well confined anatomically. So very early, those cancer cells around the ovary and fallopian tube, they can spread throughout the abdomen. And ovarian cancer is known to be a highly heterogeneous disease. And this heterogeneity is found between patients, but also within patients. And this molecular heterogeneity is observed when we compare the molecular characteristics of these different tumor deposits. And very often this heterogeneity has been associated with the development of treatment resistance, so it's clinically important, but it is very difficult or almost impossible to take tissue biopsies or to get tissue samples from all these different tumor deposits. And it's even less feasible if we want to follow this molecular heterogeneity over time. So we can't take multiple biopsies at multiple time points during the treatment of these patients. But imaging is a non-invasive way of monitoring heterogeneity, but we need a way of measuring heterogeneity. And radiomics have been shown to be or to have great potential in measuring this heterogeneity. And this is a study that showed this nicely, and the authors of this study, they computed textural features for different disease or cancer deposits in the abdomen, and then they computed dissimilarity or heterogeneity between the different sites in the abdomen, and they visualized the heterogeneity using these matrices, and the more red and yellow boxes you see on this matrix, the more heterogeneous the disease is. And this is a patient where we see only very few red squares, and this patient survived for 69 months and reached complete resection during surgery. So this was a patient with a relatively good outcome. And this is a patient where we see more yellow and red squares on the matrix, indicating higher dissimilarity, higher heterogeneity. And this patient only survived 10 months, and the outcome from surgery was incomplete resection. So just by looking at the dissimilarity between the different disease deposits, we understood that there's some prognostic information within this, and that underlines why radiomics are or can be a good tool to measure this disease heterogeneity non-invasively. However, we need to improve our understanding of the underlying biology, the biology that explains those textural features that we compute. For all these studies, again, we need a segmentation of all the disease locations. Automated segmentation makes this a lot easier and facilitates research on larger data sets as compared to manual segmentations. And this is basically our vision of an advanced imaging pipeline. We are currently working on starting not at the reconstructed images, but on the CT raw data, the sinograms that are acquired by the CT scanner. Our reconstructions are being optimized for automated segmentation and radiomics computation, and both the raw data and the reconstructed images are stored and shared via the NCITA repository. And not only we upload to the repository, but also collaborating centers, and we can share the images with our collaborators from departments like mathematics, physics, engineering, and so on. On the reconstructed images, we can then perform the automated segmentation, as you saw, and we can compute radiomics for the entire disease burden. And ultimately, we want these radiomics to be part of prediction tools on a patient-by-patient level to facilitate precision oncology or precision medicine in general. We work heavily on data integration. We do not only want to use radiomics for these predictions, but also molecular tumor data, clinical data. In the future, it will also include pathology data. And as I said, we need to improve our understanding of the radiomics features, and that's why we are performing radiomics-guided biopsies. This is an example of a custom-made 3D printed mold that we use for a tumor that is being resected during surgery, and then we section the tumor in a way that is optimized for getting access to areas of the tumor that show a particular combination of radiomic features, so that we can understand molecularly what causes the appearance on imaging. Yeah, and so in summary, I hope I could show you that we are addressing several challenges in cancer imaging with our work. We want to transition from a qualitative image interpretation to quantitative assessment, and ideally this should be as automated as possible, and that will mainly affect segmentation and the radiomics computation. We are working on integrating different data streams on a patient-by-patient level, and we are generating larger imaging data sets, particularly from multiple different centers, to then be able to show that our predictors are robust and that they can deal with heterogeneity, both in terms of image acquisition and reconstruction. And the XNAT repository is key to that, and all these efforts will hopefully lead to an improved understanding of the biology that is behind the heterogeneity in ovarian cancer that is often named as the main culprit for disease resistance or treatment resistance. Yeah, this is the end of my talk. Thank you very much for your attention. And I think we're going to take questions at the end of the session, if that's correct, or I'm also happy to answer questions now.
Okay, we can try and take a few questions now. Did you use XNAT NVIDIA integration for radiomics analysis?
No, we didn't use it for radiomics analysis. We only used it for the implementation of the segmentation tool.
Okay. And then how are you handling your pathology data? We have some projects using OMERO, QPath, but another project that wants to store it all in XNAT so they can integrate pathology and DICOM from the same location.
Yeah, currently we have not yet integrated pathology data, so we haven't transferred any pathology data to the XNAT repository. This is something that we're planning for the future, so unfortunately I can't really answer the question.
Hi Yash, watch this space, and in about 2 minutes or 5 minutes you'll find out something helpful to that question.
And then there's one last one. I have not started on all the features of ML, but it's really cool doing auto segmentation OHIF. Is there any extra setting for OHIF to work with NVIDIA?
Any extra setting? Lorena, the person responsible for implementing our segmentation tool within the NVIDIA Clara framework, is sitting just next to me, and I think she may be able to answer that question in detail. I'm just going to hand her my microphone, if that's okay.
Sorry about this. I don't know if actually Simon will comment on this in his talk later, but certainly you have now videos available in the XNAT wiki on how to install your server for your AI-assisted annotation using Clara, and how to use it, and so on. So there's information that is already available for you to do this, and I think there's probably some information in posters as well, in the poster session.